PhD, which is great. Why don't you start with your background?
Are you comfortable taking?
Oh, yeah, I can. Yeah, make that a bit easier. So hi, everyone. As Margaret said, my name is Aileen. I came to Margaret's lab in January of 2020, so right before COVID happened and all of that. So a lot of this work was done during COVID, which was interesting, but through everyone's help and support, I was able to make it to this point, and I'm very excited to share with you today my master's work that relating to diurnal variability of methane fluxes, specifically in wetland systems. Here we go. So first off, I want to start off with a couple of acknowledgements. So everyone you see listed here, my advisor, Dr. Palmer, all the members of the lab, my committee members, friends and family, and in particular, I want to shout out to Alec Armstrong, who without his help, none of this would have been possible, because he was out there with me every second of sampling this past summer to get all of this together and get all of this done. So also big thank you to Dr. Yarwood's lab as well for letting us use her chambers throughout this study as well, which was great. So really to get right into it, and we're going to give a little background about wetlands and methane. So why study wetlands? Why study methane emissions from wetlands in particular? So wetlands are great for, yeah. So wetlands are really great for carbon sequestration because they can store a lot of carbon for very long periods of time. But they're also looking at this figure on the right here. They're also very large sources of methane emissions as well. Up to, throughout a single year, they can generate as much as 80 to 280 teragrams of methane per year. And it's also, if you look at this figure down here, seeing the largest increases in atmospheric concentrations as well. So it makes it kind of concerning considering global warming potential. It has a global warming potential 25 times greater than CO2 over a 100-year period. And furthermore, there's evidence that anthropogenic climate change may lead to large feedbacks from wetland emissions, especially in terms of methane. So it's made it very important to accurately measure and model what these emissions will look like and how these systems will be impacted. But despite this need, there's still a lot of uncertainty in flux estimates, and because of a lot of spatial temporal variability that exists in fluxes. So this spatial temporal variability is driven a great deal by very complex and codependent relationship between a lot of influencing abiotic and biotic variables. And ultimately, it's the balance between the production, the consumption of methane by methanogens and methanotrophs, and then the transport of methane out of the system that will govern the magnitude of fluxes that we see. And all those in turn are influenced by a lot of variables like the hydrology of the system, what the temperature is like, how much dissolved oxygen is available, and things like that. And these conditions in turn will determine how much flux is actually transported out of the system via these different pathways that we see up here. So like a big one is plant-mediated transport through wetland vegetation's parenchyma, ebullition as well, so like any bubbling of fluxes out of the system. It's a very fast method of transport. And also diffusion and hydrodynamic transport. So just molecular diffusion out of the system. And also any advective processes like vertical stirring of the water column versus by wind-driven stirring or temperature differences in the water column, creating thermal convective stirring with that. So this in turn, these relationships between all these variables can lead to a lot of spatial variability that we tend to see in wetland emissions, and it can lead to a great deal of heterogeneity across a single wetland system. So this can be categorized by breaking up the system into a series of different patch types, so to speak. And these patches can be variable by their topography, their soil chemistry, their hydrologic regime, and the different vegetation communities. Like in this picture alone, you can see a couple of different patches of different dominant vegetation types right there. So this can lead to characteristically dynamic fluxes within a single wetland system. With that, there we go. So for this study going forward, when I refer to patches specifically, I'm going to be talking about patches that are defined by a dominant vegetation type. So whether we, like right here, we have a lot of emergent vegetation or primarily submerged vegetation with that. So these changes in these processes and rates can also lead to a great deal of temporal variability of fluxes. And in regards to temporal variability, there are some aspects that have been very well documented. So a lot of systems will see pronounced seasonal variability in fluxes. So like during the growing season, right here, we tend to see much higher fluxes and a much higher magnitude of fluxes compared to other parts of the year. But other aspects of temporal variability, like diurnal variability in particular, like short-term variations in short-term patterns of day-night over day-night cycles, still requires more focus. And the reason for this is because nighttime fluxes haven't been as well monitored as daytime fluxes have been in these systems. And one of the reasons for this, which I'll get into a little bit later, is that because of current methodologies that exist, a lot of the time we have to discard nighttime data. So we would expect to see, to some extent, some diurnal variability in these fluxes because of all those environmental variables that we just talked about that influence fluxes. Some of them tend to undergo DO patterns undergo DO patterns themselves with that. And there's also, regarding diurnal variability, another reason why more research is needed is because there's mixed evidence in literature. So in some studies in some sites, there have been examples of recorded DO patterns, like this study by Duan et al. They found very clear, whoops.
Okay.
Yeah, thank you. Very clear patterns of DO variability from a Phragmites species in one wetland. And then there are other systems that don't have DO variability, like in this study by Milberg et al. It's kind of all over the place. There's no clear pattern with that at all. Surprisingly enough, this was also from a Phragmites species, but in a different wetland. So these two studies in particular really emphasize this idea that we can't make broad generalizations about diurnal patterns and fluxes because these fluxes might be very system specific. And to take a step further, going back to what we just talked about, spatial variability, these patterns and maybe patch specific as well, depending on the conditions and the main transport mechanisms that exist in each of these patches. Okay, I'm gonna hide this again. So I mentioned in the previous slide that one of the reasons we don't, there are some issues with the current measurement techniques, and it's one of the reasons why we don't have, why nighttime fluxes in particular haven't been as well monitored. And that's because, okay, here we go, getting there. One of the main methods that exist currently are the use of eddy covariance flux towers, which are really great because they allow for continuous measurements over a very large footprint area, but a lot of the time they're nighttime. The nighttime data is often removed from the data set because it's not as reliable as daytime emissions. And on the flip side, yeah, they allow for continuous measurements and nighttime data is often removed. On the flip side, with chamber-based studies, these are fantastic when you want to look at small scale variability. So like over different patch types. And like you want to look at individual differences across a single wetland system. But the measurements can often be very tedious and time consuming to record. So many studies rely on scaling up a single measurement taken during the day to represent 24-hour fluxes. And another common assumption made in a lot of these studies is that plant-mediated flux tends to dominate total methane fluxes. So many studies only focus on the contributions from emergent vegetation. Both of these methods might actually be bias estimates of fluxes. The scaling method assumes that there's very limited diurnal variability in these emissions, and the plant-mediated flux and focusing just on those types of patches might ignore the contributions made from other patches of vegetation. And this is a problem. Like, one of the problems with these assumptions is really highlighted with a couple of more recent studies, like a study by Godwin et al. And this one by Poindexter et al. And this one, like I said, is the Poindexter et al. Both of these studies suggest that hydrodynamic transport and temperature-driven stirring of the water column is actually contributing more flux, contributing a lot to the DO variability of methane fluxes than originally thought. And so, like you can see right here, the temperature differences in the water column throughout a day-night cycle and the subsequent stratification of it and breakdown as it cools down. Of that stratification as it cools down at night can lead to pulses of flux that you wouldn't see during the day. So this study in particular, going back to those assumptions that we just talked about, if you're just focusing on the contributions from emergent vegetation, you would be missing this contribution. And if you're only sampling once during the day and then scaling up those fluxes, we'd also be missing this potential contribution as well. So to address both these assumptions and also to help contribute to the knowledge gap that exists in DO variability of methane emissions in these wetlands, we performed a study in a restored seasonal mineral soil wetland on the Del Mar Peninsula in Maryland, USA over the summer of 2021. The main objectives of this study were to determine if there is variability in methane emissions over multiple DO cycles. So is there DO variability? Does this DO variability change if you're measuring from different patch types? So how does it compare against these, between these dominant vegetation types? And also more generally, which environmental variables are drivers of DO variability, and kind of looking more specifically based on those previous studies that I just pointed out. Is DO variability at all associated with thermal stratification and convective turnover? So, for moving on to our actual study and the methods that we did, these are aerial photos showing the wetland that we worked in. So this wetland was in the Choptank River watershed in Queen Anne's County, Maryland, on the Del Mar Peninsula. So that's Del Mar Peninsula right there. It was a restored wetland and 1.33 hectares in size. And it was ditched and drained back in the 1980s and converted to pasture land. It was restored in 2003 by plugging the ditches and cutting down all the woody vegetation. And it's classified as a freshwater emergent wetland. So as you can see, these are some ground photos of the wetland site that we worked in through various stages of the year. And it's characterized by a lot of vegetative diversity, which you can see here. So a lot of different vegetative patch types. And like I said before previously, when we're talking about spatial variability, these can be broken up into a series of different patch types depending on the dominant vegetation type. So we have grass mats are a couple of dominant ones. We have sedge tussocks, which you can kind of see right in here. There's low growing emergent species, which you can kind of see scattered all throughout in here. There are areas of submerged aquatic vegetation, and there are areas of open water. So moving on to our own patch selection, this is a aerial map that was put together by a member of our lab to showcase how patchy this wetland can be, for lack of a better term, show the heterogeneity of it all. So each of these colored spots represent a different patch type with this. These red bars are boardwalks that we installed, and these gray represent different chambers with that. Thank you. So from those patches that we just talked about in the previous slide, we chose 3 different dominant vegetation types to represent different types of vegetation cover. So we had our emergent veg patch, which is dominated by Carrick species. We had our submerged aquatic vegetation patch, which is dominated by Juncus repens, and we had our no veg patch over here, which had no vegetation. And it's important to note that because of these different types of vegetation cover, all 3 of these patches are most likely dominated by different transport mechanisms of fluxes. So like this patch, the emergent veg is probably dominated by emergent vegetation, plant-mediated transport. The sub-veg may have some plant-mediated, but it wouldn't be a direct conduit to the atmosphere, it would just go to the water column. So this one would probably be dominated by diffusion, and ebullition, and hydrodynamic transport. And the open water patch would just be ebullition and hydrodynamic transport / diffusion with that. And like I said, the boardwalks were installed to prevent disturbance while sampling with that. And this is our walking path that we used for all of our sampling. So for our campaigns, we had 3 campaigns over the summer of 2021. They were June 20th to the 25th, July 26th to the 30th, and September 12th to the 17th. And there were 2 sampling days approximately, the 2 sampling days in each campaign, and gases were sampled 6 times over a 24-hour period. The approximate sampling groups were 9 p.m., 1 a.m., 5 a.m., 9 a.m., 1 p.m., and 5 p.m. And here are some pictures. These are all the files we use for sampling, and here's some pictures of setup in the wetland. This was at like 8:55, right before go time on the first day of sampling. Okay, and for our chambers in gas sampling, the floating chambers that I designed and built, you can see right here, were used for sampling in the submerged aquatic vegetation and the no vegetation patches. And these were made from small aquariums, and then which we attached a little PVC pipe floats to, so that they would float like here. And we used static chambers, courtesy of Dr. Yarwood's lab. This is an example of a schematic of those in the emergent vegetation patch. And collars for these, like the ones you see right here, were installed a month prior to sampling in the site to prevent, so that any disturbance we caused while installing these, the site would have time to revert to normal conditions. Sampling always began in the emergent veg patch, and it would subsequently move to the sub and no veg patches. And all sampling was done manually with 30-minute incubation times. And samples were taken at 0, 10, 20, and 30 minutes throughout that incubation. And gas samples were eventually analyzed using gas chromatography for methane and CO2 concentrations, but in this presentation, I will only be presenting the methane results. So emission rates were corrected with measurements of chamber volume, atmospheric pressure, air temperature, and flux was calculated as a linear function of gas concentration throughout the incubation period. So in addition to this, I was also, I had a series of physicochemical sensors that were placed to analyze water pH, water temperature, and dissolved oxygen concentrations, and these were patch specific. They were on a series of little sensor stations like this one. It's a little diagram of what it looked like. We also had a flux tower on site, so we were able to get measurements of wind speed, PAR, air temperature, air pressure, and relative humidity as well for that. So we did this as a way to pair these flux measurements with these environmental variables to help understand what might be driving these patterns that we might see with that. In addition to the ones I just talked about, we also had measured water depth using wells on site. And in the emergent vegetation patch only, we measured stem count. So moving on to the results for this. So to like kind of set the stage and give you an idea of what summer conditions were like in this system and what the flux results were like. Based on that, we're going to start out with the physicochemical results. So to start out, water depth, you can see in this table here, water depths in each of the patches throughout each of the 3 campaigns of the summer. And the big takeaway from this chart is that the second campaign had the lowest water depth in each of the 3 patches. And then it jumped up again, moving into the third campaign. But water depth was always shallowest in the emergent vegetation patch, and it was always deepest in the no vegetation patch. And here we have a range of air temperature throughout each measurement occasion. For the most part, there wasn't too much variability in the air temperatures across sampling. But the biggest takeaway from this is that on the June 23rd and 24th day of sampling, a cold front moved in, and it was really cold out there. Here on the right, these are some examples of some of the environmental variables that we measured from the flux tower. So here I'm only reporting, because there's a lot of variables, there was a lot of campaigns, I'm only reporting campaign 1 to give you guys an idea of what these look like throughout the summer. And the big takeaway from this is that air temperature, the PAR, relative humidity, and wind speed tended to undergo some form of DL variability and had very distinct DL patterns. And you can see here, almost like cyclic patterns. But air pressure, while variable, did not undergo this DL variability, but it was very variable throughout the sampling times. And these shaded areas represent the the days that the approximate times we were out there sampling. In here as well, these one, like I said before, the water pH, water temperature, and dissolved oxygen saturation, which you can see down here, were patch specific. So for this instance, I'm only reporting, to again, give you guys an idea of what it looked like. I'm reporting the results from the no vegetation patch from campaign 1. So this is pH. The red line represents measurements just above the sediment water interface. And the blue lines represent measurements just below the water surface. And as you can see, the water temperature and dissolved oxygen saturation also tended to undergo DL variability with that. Okay. So again, to set the stage, I'm going to be moving into the flux results that we got. But prior to doing that, I wanted to define for you guys what exactly we were looking for with this and what exactly a DL pattern is. So based off of literature, and it was the Milberg paper in particular, for this study and for the results of the study, we're defining DL pattern as a recurrent peak or low in fluxes. The low got cut out, but peak or low in fluxes recorded at the same time over multiple DL cycles. So we're going to be starting out with the emergent vegetation results for this. And these are a series of what our flux results look like for our June campaigns, and 1 day in our July and 1 day in September campaign with this. So for all of these results and analyses, I ran a repeated measures ANOVA to get an idea of how different these fluxes were over the time periods. And as you can see here, there was DL variability exhibited, but these differences were not significant, and there was no consistent pattern across all of our campaigns. They were kind of, in a way, it was very inconsistent what we were seeing. And mean flux tended to be around 19.8 milligrams of methane per meter per hour. Mean minimum flux was around 10.94, and mean the max flux was around 31.03 for the entire summer. So based on these measurements and based on how much, again, calling back to our one of the assumptions we were trying to address, whether or not this midday sampling or sampling throughout the day would be representative of 24-hour fluxes, can conclude that, like, on average in this emergent vegetation patch, midday sampling is usually a good representative of 24-hour flux. Okay. And moving on from this, like as a way to combine what we found in the, combine our flux values that we found and combine the environmental covariates to see, kind of get a good idea of like what is controlling and driving fluxes, these patterns that we see in systems. Started out with a very basic correlation analysis just to see what might be what of the environmental variables that we measured might be related to these fluxes. So like I said, this was just a preliminary analysis to get a good idea of what was going on. So these were the variables that we measured that had a correlation with the emergent vegetation fluxes. And air pressure and stem count, surprisingly, had a negative correlation with fluxes. And water temperature and air temperature had a positive correlation with that. So to take it a step further and get a better idea of like how, because like I said, that was a very preliminary analysis. That was just looking at individual variables matched with the flux values. Wanted to take a step further and see how potential interactions between these variables might be responsible for the potential patterns that we saw and the emissions that we saw. So I used a forward and backward stepwise progression selection to put together a series of models for this data and to examine how, like I said, how these potential interactions variables might influence flux. I also used a dominance analysis to determine the relative importance and contribution of each predictor variable and get an idea of which variable kind of dominates over another. So this was the finalized model that I came up with for emergent vegetation. It includes the timing of sampling, the chamber replicate, water temperature, air pressure, relative humidity, air temperature, and stem count. And in running the dominance analysis, I found that sampling time, water temperature, and air pressure tended to be the dominant predictors of the fluxes that we saw in this wetland. And I want you guys to keep in mind what the dominant variables in this patch is, because I feel like I think it highlights the idea that in looking at these patches individually, we might find different patterns and we might find different relationships between these variables. So we're moving on to the sub-veg patch and looking at the deal patterns that we found here. So again, these are examples of the fluxes that we saw in the measurements we took. And as similarly to the previous slide in the previous patch, the emergent vegetation patch, you can see that there was definitely deal variability exhibited, but unlike the previous one, there were consistent peaks at 1 p.m. that were repeated throughout each of the sampling days. So going back to our definition for deal patterns, this is a recurrent peak over multiple, at the same time over multiple sampling days. So we can call this a deal pattern in emissions. And also another point to make is that daytime fluxes, so fluxes that occurred during the 9 a.m. to the 5 p.m. time, usually tended to be higher than the nighttime fluxes. So any fluxes from 9 p.m. to 1 a.m., 5 a.m. So on average, based on this and looking at what the mean flux tended to be, which was around 17.54 throughout all, like for a daily 24-hour flux, and comparing that with what mean flux tended to be at this 1 p.m. time, again, going back to the one assumption about sampling during a 24-hour period and scaling that up, we see that this, using a measurement like this, might not be a good representative of what 24-hour flux actually is, because looking at these numbers, mean flux at 1 p.m. was anywhere from 46.7 to 67.5 milligrams of methane per carbon per meter squared per hour. And comparing that to what the daily mean flux tended to be, that was around 17.54. So in this patch specifically, using a midday sample might actually be an overestimate of what fluxes actually were. Okay, so again, to get an idea of like what variables might be responsible, or environmental variables might be responsible, started out with a very simple correlation analysis for this. And these were all Kendall correlation tests for analysis to see what could potentially covary with this. And from all the environmental variables that we measured, these are the ones that I pulled out as significant. So for these, pH, water depth, and air pressure tended to negatively correlate with flux values. And water temperature, air temperature, wind speed, and air temperature tended to positively correlate with fluxes. So again, going based off that, I put together using a forward and backwards stepwise selection process to see what would see what variables would best predict what the fluxes were. I ended up with this finalized model, which is a lot more complicated than the last one. The variables included for that were sampling time, the campaign, water temperature, water column, dissolved oxygen concentrations, air temperature, air pressure, relative humidity, and maximum wind speed and PAR. And from this, after running the dominance analysis, sampling time, water temperature, and air pressure tended to be the dominant predictors of this model. And again, going back to what we found in the Emergent Veg patch, you can see that it was a different series of variables are the dominant predictors in this model of the sub-veg dominant predictors for flux. And again, going back to the idea of some of the differences between these patch types, they're both use different types of transport to get methane from the soil to the atmosphere with that. And moving on finally to our no vegetation patch. These are, again, some examples of what our fluxes look like. And as you can probably tell, looking at these axes, fluxes were very, very low comparatively in all of our patches, in this patch compared to our other patches. Excluding our July sampling, that was the only time when we saw any significant contribution of flux. So that was happened to be when the water table was lowest. And like I said, we saw diurnal variability exhibited in these flux patterns. And again, we tend to see these peaks at 5 a.m. consistently across multiple sampling days. And we tend to see these lows at 1 p.m. across the sampling days as well. Yeah, like I said, the flux was very low, if not negative, except for the July sampling. And on average, mean nighttime flux was larger than, looking at this July sampling as well, the mean nighttime flux, so fluxes from 9 p.m. to 5 a.m. tended to be larger than the 9 a.m. to the 5 p.m. sampling. So again, calling back to one of the assumptions we were trying to address in this presentation, the idea of sampling once during a 24-hour period and scaling that up, looking at these measurements throughout a 24-hour period, mean flux tended to be around 1.37 milligrams of methane. And the mean flux at 1 p.m. usually tended to be lower than that. So again, midday sampling may not be a good representative of a 24-hour flux estimate. It might actually be an underestimate of flux. And again, did a series of correlation analyses to see what environmental variables might be responsible for these patterns that we saw. And for this patch, water depth and air pressure tended to be negatively correlated with flux. And the temperature in the water column and relative humidity tended to positively correlate with flux. And looking at our model, this was the finalized model that I settled on. It was based on AIC scores. This one had the lowest. And it included water temperature, water depth, and wind speed as the best predictors of the fluxes that we saw. In the dominance analysis, water temperature and water depth were the dominant predictors of that. And again, going back to the slide that we just looked at and what the patterns that we saw, the only time we saw a dominant contribution of flux in this patch was during the July campaign when that water depth was actually the lowest. So it makes sense that it was included in this model. Also, this wasn't a significant correlation, but again, going back to this idea of hydrodynamic transport and how temperature-driven stirring of the water column might lead to contributions of fluxes. It wasn't a significant correlation, but this 5 a.m. peak in this no vegetation patch consistently happened to coincide with times when the water column was either the top and the bottom of the water column were either the same temperature, or the top of the water column was actually cooler than the bottom, which I thought was pretty cool. But again, it's not a significant relationship. Moving on, for our did a patch comparison of all the patches and all the fluxes that we saw. And these were all compared, like I said, using a nested repeated measures ANOVA. So this is our first campaign results. The red colors are the emergent vegetation patch. The green are the no vegetation patch, and the blue are the submerged aquatic vegetation. So this was our June 20th to 21st sampling, and consistent. The ultimate takeaway from this is that consistently throughout the entire summer sampling, the no vegetation patch had significantly lower fluxes than the emergent vegetation patch. And looking at this one in particular, the big takeaway from this one is that consistently the sub-veg patch had more fluxes that were more comparable to the emergent vegetation patch. These two patches had significantly higher flux than the no vegetation patch. When we move on to our July sampling, when the water table happened to drop, there were no, excluding this 1 p.m. sampling, there were no significant differences in the fluxes between any of the patch types. So no veg, sub-veg, and emergent veg were all comparable in flux magnitude throughout this. Then moving on to our final campaign, it was interesting. The submerged aquatic vegetation patch and the no veg patch flux magnitudes were actually more comparable than they were to each other than they were to the emergent veg patch. The emergent veg patch had consistently significantly higher fluxes compared to these two patches. So again, big takeaway from all of these figures put together is that beginning of the campaigns, sub-veg was more comparable to the two patches with some form of vegetation, or more comparable to each other. Then they move, when you move to the middle of the summer and the middle campaign, all the patches were comparable in flux magnitude to each other. And then moving on to the final one, only the emergent vegetation patch. The emergent vegetation patch had significantly higher fluxes than any of the others. Okay, so I'm still working through a lot of this data and kind of putting together my own thoughts in regards to these patterns that we saw and kind of getting my thoughts together with it. But some conclusions that, and some things that have stuck out currently with this are that flux measurements and evaluations of DL patterns and DL variability should be measured based on the dominant vegetation type, or the lack thereof of vegetation. And also going back to that one assumption with chamber-based studies about scaling up measurements, depending on the patch type that you're measuring from, midday sampling might not be the best representative of what the average 24-hour flux is, depending on the patch type you're looking at. Also, in regards to potential future work, one thing to note is that, and going back to looking at our environmental variables, specifically the water table, the water table did not change too much, and we didn't undergo, this sampling didn't take place during a series of changing hydrologic conditions. There was always some amount of standing water in each of the patches. So it would be interesting to continue this work under a series of changing hydrologic conditions. So what happens when the water table completely drops? What happens when there's no standing water in these patches at all, and the soil starts to dry up? So it'd be interesting to see how that affects patterns that we see. Also, these results may be system specific. So again, this study and this research highlights the need for more studies that focus on DL variability and fluxes from a range of wetland types and vegetation patch types and cover. And so that is the end of my presentation. So thank you for listening.
Air pressure looks important.
Yes, air pressure is also important.
So I'm curious, so that one date that you had the second sampling panel basically is the one where you had the cold front come in, right?
Yes,
And so,
yeah.
What happens if you exclude that date from some of these models? Because it seems like air pressure is there, but is air pressure really driven by the fact that you had that cold front basically in that second sample?
That's a really interesting point that I did not follow up with. I want to follow up with that now in looking at everything, because that is not something I explored or tried to do with that, because my thinking right now and why air pressure was important in each of these systems with the transport mechanism specifically is for emergent vegetation. At least in some reading that I've done, it seems like that can have an effect on how well it's transported through the plant mediated transport acts basically, and how much methane it can transport. And going over to ebullition and diffusion, ebullition at least, there have been, if I'm remembering correctly, there have been some studies that have noticed with decreasing air pressure that might lead to, like that pressure differential will lead to better transport of and faster transport of methane out of those systems via those pathways. But yeah, no, that's a fantastic follow up to these analyses that I have not explored yet, and I would like to now, because that's a good question.
Yeah, no,
Yeah,
it is interesting, and I think there is a break point where they have shown that air pressure basically starts to impact that or not, and I can't think of the magnitude of that, but it's definitely something I think that maybe look into. So.
Is that on the plant stuff or on the evolution?
I think it's on the evolution, which makes it really interesting that you see it even in the emergent vegetation piece. That part of it actually strikes me the most, because my understanding is that's all water column dynamics. But that still, of course, could be affecting in that emergent vegetation also. So yeah.
But it might be super important in the sub-veg if it is primarily on the evolution, because that's where evolution is.
Yeah, especially like in going back to emergent veg and the patterns that we saw, one of the biggest surprise, that was the biggest surprise to me in doing this research. Especially because of plant dynamics and maybe model opening and closing, I kind of expected there to be more of a very significant deal pattern. But to see there was basically almost constant transport to some magnitude throughout a 24-hour period, which at least when going into this research, when I was thinking about emergent veg, I was like, this one's definitely going to have a deal pattern. Like I'm expecting this. And then I found that, well, it's actually more of a constant transporter of methane throughout 24 hours, which I thought was cool. Yes.
Can you kind of following up on that statement? I mean, as you showed that first kind of plant and you showed where methane can come up, I mean, looking at the results that you got, looking at without vegetation and looking at, you're seeing more of a constant flux. Do you have a feel, or was your research able to get at where most of the methane is being emitted? Is it from the sediments? Is it from the plants? Where are you able to state that with the results that you have?
With my results, no, I'm not able to make, especially like in the emergent vegetation patch, I'm not able to make that. I primarily been assuming with this that plant mediated transport is the dominant transport mechanism in that patch specifically. So I'm not able to say whether it's definitely coming through the plant system, the plant itself through plant mediated transport, or if it's more soil respiration, direct soil respiration with that. I would honestly be interested in looking at the model conductance of these plants going forward as a way to kind of potentially look at that and get a better idea of how much gas movement there might be through the plant stomata. And if I think that would give a better explanation, specifically in the submerged aquatic, not submerged aquatic, the emergent vegetation patch. What if is plant mediated transport definitely dominating transport in this patch type. Does that answer your question with that, or?
I'm just trying to get at the thought, I mean, from kind of the bacterial level and methane.
Yeah,
production
yeah.
in Wetlands where you have anaerobic sediment. I'm just trying to get a handle on the percentage that's coming from.
Yeah, yeah,
Sediment, what's being transported just straight through the sediment,
Mhm.
what's coming through emergent,
yeah.
and how you can, you know, with the 3 patches that you have, how you can
Mhm.
kind of contribute to the knowledge of
Like each of the
where
dominant,
methane's
yeah,
coming,
yeah.
where it's being produced
Mhm.
and where it's, how it's emerging out of this environment.
Yeah, but one of the, I think, again, and this is an assumption I had throughout my entire study, was that in the patches with emergent vegetation, what you see a lot in literature with this is that emergent vegetation, if it is there, it does tend to dominate fluxes, like anywhere from 30 to 100%, I believe is a percentage I've seen in some studies. Flux will be directly through, directly plant mediated transport. Looking at the other patch types, submerged aquatic vegetation, in my presentation, I said that we are assuming that hydrodynamic transport or ebullition would be dominating, specifically because the submerged aquatic vegetation is not a direct conduit to the atmosphere like emergent vegetation would be. Like if it is transporting methane, it's just going to be going to the water column. It's not popping up out of the water column. So in that case, if there is plant mediated transport going on, it's not completing the transport. And then in the no vegetation patch, there's no vegetation there present to potentially contribute. So those ones are definitely, I don't have a good estimate of which is ebullition versus diffusion or hydrodynamic transport in those patches, but kind of lumping them together. It's like those are the transport mechanisms that are responsible in that patch type.
So any differences that you saw between the submergent vegetation and the no vegetation, could that have been horizontal transport? Like, you know, I guess because they're all linked, how do you kind of make the distinction of where the methane?
That's a really good question, and one that is not. I think one of the kind of pitfalls with these types of studies is and especially something that I need to think about more, is like I always forget about that potential for lateral movement. Been thinking of this very much on a vertical movement. The production that's going on is directly coming up, but there is that potential. So for lateral transport with that. For the big differences that we saw in the fluxes, specifically between submerged and no veg, my thinking with that has been the potential for plant effects. The fact that there is vegetation there, they're not just they can contribute to transport out of the system, but there's also priming effects that could be going on, like they're providing organic matter for these microbes to use. They could be providing root exudates with that. So another follow-up to this study would be really looking at the individual plant effects that these types of plants specifically are having.
So one, just quickly on that, what's the sampling rate of the thermistors? Fast?
Oh, the thermistors, the temperature, the temperature pendants, they are 5-minute intervals.
That's as often as it samples?
That's what I had it to. I would have to look to see if it's more rapid with that.
I was going to say, if it sampled rapidly, you could take a difference in temperature changes as an indication of shear.
Yeah, when you're sampling, you're only getting the quality, the quality of the biogas, the percent of methane versus percentage CO2, not how much biogas goes into the chamber. There's not a.
So the sampling is it's measuring the concentration in that headspace of the chamber. So like how that concentration starts, like one of the reasons we take as like a 0-minute sample is like this was the starting concentration of these gases in this headspace. And then the subsequent ones are to see how is this changing over a over a 30-minute incubation, to get an idea. How these concentrations are changing and how that's like a linear regression of those concentrations can be used to calculate what the flux is out of that, into or out of that system. Yes.
So Aileen, I enjoyed your presentation. So a lot of your discussion seems to be focusing on sort of transport mechanisms out of the soil sediment.
Mm-hmm.
But are you operating on the assumption that the amount of methane generated in those different patches is similar? I mean, you alluded to the fact that where you've got emergent vegetation or submerged vegetation, you may have greater carbon inputs than where you don't. And we would expect that where you have emergent vegetation, you get much, much higher carbon content to those soils, sediments.
Mm-hmm.
And so I know are there any sort of operating assumptions on this, or are you recognizing that it's really kind of a combination of generation and transport in these?
It is
systems?
that is an important point to make. And I think it's one of the reasons we need to highlight that these are coming from different patch types, because I don't know how much is being produced. That was not something that was measured. So ultimately, like you said, I'm highlighting the transport of these fluxes out of the system, but that's not an indication of how much is actually being produced or oxidized as well. I'm only seeing one piece of the puzzle. So that is something I will need to highlight with that. It's like, I'm highlighting one piece of the puzzle with this. This is what transport out of these different patches look like, but it doesn't address.
So of course, as a soil scientist,
Yeah,
I
yeah.
like the idea of looking at what those materials are,
Yeah.
and to get some sense of what we might anticipate from the.
Yeah, no, it's something that I've been thinking about in terms of PhD work as well. Following up, what are the plan effects of these systems, and how can that impact what we're seeing in the flux measurements and transport? Because that was, aside from saying like, yeah, these are different vegetation types and different patches of vegetation, my work did not focus on what's happening. They're below ground impacts, other than just saying there might be these differences depending on what the vegetation is.
That's
Yeah.
part of Sean's work.
Yeah.
but,
Yeah.
I'm not positive what he's got. Yeah.
Mhm.
And don't lose the point that really consumption, I mean, in some of these, consumption drives it much more, I think, than production.
Yep.
So in residence time and the soil,
You
So-
know,
to be able to
oxidizers?
yeah, the oxidizers.
But my point is, of course, in emergent vegetation, we anticipate much greater carbon content in those systems to begin with than you get
Yeah.
in. the
Hey, but it depends on what the exit is and where all sorts of things.
Were you able
There's
to
no-
sort of
there's no
compare and
general-
contrast different types of plants? For example, like, did your study include any cattails or phragmites?
No, it was only one. One of the reasons why it was submerged aquatic vegetation, it was all carex for emergent veg and no vegetation. It was really the functional difference. Like, is this emergent vegetation versus submerged versus no vegetation, rather than looking at individual plant differences with that in the different functional groups.
One last quick question before we have to vacate the room. for
Yeah.
5
You did note different times of day where you had the peak methane fluxes between the vegetation patches, but then you also mentioned that there was somewhat consistent fluxes of methane in the emergent vegetation. Do you know why you saw these differences? Or do you have like thoughts on why you saw differences in time of day?
Why I saw that variability
And
with
why
that.
different peaks of methane, and is that really?
So,
1 minute
yeah.
response.
Okay, so for the for the emergent vegetation specifically with that, that's a really good question. And I think does come back to this idea of emergent vegetation and plant mediated transport may happen at different rates. Like, I don't have depending on different environmental conditions. So maybe in some way related to air pressure, what the relative humidity might be, and some environmental effects like that might be one of the reasons for that variability with that. But more so, one of the reasons I wanted to highlight that there was, despite that variability, the mean fluxes tended to be around the same average despite that, so was a big thing I wanted to highlight with that. I don't think I answered your
Well, we're gonna have to
Yeah,
bring it to a
sorry.
close, as we have a class
I can
coming in
talk to you
behind
more
us.
about it.
Thank you very much.
Yes.
Dave.